In the second part of Chapter 5 recording, we will discuss further extensions from the Mendelian inheritance, and we will start with sex influence and sex-limited characteristics. In previous chapter, we were discussing traits which were encoded by genes located on sex chromosomes. Those are sex-linked, located on sex chromosome, sex-linked, located on X chromosomes, X-linked, located on the Y chromosomes, Y-linked. Here we will talk about the inheritance of traits which are encoded by genes located on autosomes. However, their manifestation is affected by the sex in which you find them. So we will start with sex influence and sex limited characteristics. So sex influence and sex limited characteristics, both, as I mentioned before, are encoded by genes located on autosomes. However, the expression differs in males and females. Let's now look at some examples. One of the best examples for sex influence traits is inheritance of beards in goat. Beard is encoded by allele which will be labeled B. So this allele, although inheritance, respectively, let me step back, inheritance of these traits follow Mendelian principles. However, it is the behavior of this allele which is different in males and females. It does act as dominant allele in male, but recessive in females. Why it is possible, or how is it possible? It is because many of the manifestation of many traits which are influenced by sex are actually influenced by hormonal levels, by hormones which are different in males and females. So let's look at this example. For example, we already said that allele encoding beard is dominant in males and recessive in females. So, for example, if you have homozygous, which carries both allele encoding beard, both males and females will have beard. They will be bearded. If you have individual who does not carry any of the allele encoding beard, so both males and females will be beardless, no beard. However, where you can see, or you will see difference, is when you follow a heterozygous, which carries one allele encoded beard. Now, here you will see difference in males and females. So in males, because this allele act as a dominant, so the trait will manifest. So all males which are heterozygous will be bearded, yes. However, in females, this allele acts as a recessive, or manifests itself as recessive. So all females that are heterozygous for this allele will be beardless. Yes, beardless. So this inheritance is possible because appearance or manifestation of certain traits is under control of different hormones and different pathways, which are regulated differently in males and females. Now, not all sex-influenced traits are dominant in male and recessive in female. It might be reversed. It depends on individual case whether the allele would manifest itself as dominant or recessive. However, what is the most important thing is that sex-influenced traits, although following Mendel's principles of inheritance, are exceptional because their expression differs in males and females. Sex-limited characteristics are also encoded by genes located on autosomes. However, contrary to sex-influenced characteristics, sex-limited characteristics are only expressed in one sex, either only in male or either only in females. Now, here is an example, so-called cock feathering. Cock feathering is an autosomal recessive allele which is manifested only in males, and it gives this very particular phenotype which you can only see in male, in cock. So basically, if you have individual which is homozygous recessive and it is a male, it would have this particular feathering. If female is homozygous recessive, like one outline here, right here, she would not have this so-called cock feathering, but she would show hen feathering. So basically in female, this phenotype does not occur. So it is limited only to males. Now, in humans, there is very similar male-limited characteristic which is called precocious puberty. However, precocious puberty is autosomal dominant and it does manifest only in male. What it is so-called premature puberty, which appears in boys before the age of 4 and is manifested by enlarged penis, voice deepening, and pubic hair developments in very early age. So if male or boy is heterozygous, or if it's heterozygous, or if it is homozygous dominant, of course, it will show precocious puberty. In female, it does not manifest. So female can be heterozygous, and if female is heterozygous for precocious puberty, she does not show any phenotype. Even if she is homozygous, she does not show any phenotype, but she can pass it on her sons who will manifest precocious puberty. Sex also influences inheritance of traits which are control or mechanisms of inheritance follows the cytoplasmic inheritance, genetic maternal effect, or genomic imprinting. So first let's talk about genetic maternal effect. It's a very particular mode of inheritance in which the phenotype of an offspring is determined by the genotype of the mother. So not by its own genotype, but by the genotype of mother. Usually the traits or characteristics which arise or start to develop very early after fertilization are controlled by this mechanism. And it's because the protein, respectively substances which are present in the cytoplasm of an egg, control development of these traits. You recall that upon fertilization, egg provides genetic material and cytoplasm. Sperm only provides genetic material, so early development is regulated by proteins transcribed in cytoplasm derived from RNAs of maternal origin. It's why the traits which start to arise very early during development are very often controlled by genetic maternal effect. And you have one example right here, which is shell coiling in the snail Limnia peregra. I hope I pronounce it correctly. So in this case, the shell coiling, dextral coiling, is dominant. So if you cross male here, which is homozygous dominant, with female, which is homozygous recessive, you get offspring which is heterozygous. So this since dextral is dominant, if this trait follows Mendelian inheritance, its offspring would be dextral. However, since the shell coiling is controlled by the substances present in maternal cytoplasm, this offspring manifest maternal phenotype, and it has sinistral coiling, shell coiling. If you let self-fertilize this individual, you will get a ratio of genotype 1/4 homozygous dominant, 1/2 heterozygous. So 3/4 should have dextral shell coiling. However, 1/4 is homozygous recessive. So again, according Mendelian inheritance, it would have sinistral coiling. However, since it's a maternal parent which determines the phenotype, all F2 generation will be dextral. So keep in mind, genetic maternal effect means that the phenotype of an offspring does not reflect its genotype, but the genotype of the mother. Genomic imprinting is another example of traits, characteristics which are altered or affected by sex of the parents. In genomic imprinting, the expression of gene is affected by its parental origin. What does it mean? If an allele for particular gene is inherited from father, it manifests itself differently than the same allele inherited from mother. One of the best examples is inheritance of insulin-like growth factor 2, which controls or contribute to the growth of embryos. This gene is active in human and in mice, so many studies were done in mice development. For this particular gene, only allele which comes from father, meaning parental allele with paternal origin, is active. Maternal allele, meaning allele which comes from mother, is silenced. So it's not active. So basically, the size of embryo is determined by this combination of one active allele, one inactive, but both have the same sequence. So how is it possible? What does control it? Epigenetic changes. In this particular case, the methylation of DNA alters or suppresses expression of Ch5.2 allele inherited from mother, while you don't find this methylation pattern on the paternal allele. We will talk more about epigenetics later on. To understand genomic imprinting, you have to realize that epigenetic changes which do not alter sequence, can alter gene expression, and this is one of the case. Now, there are another characteristics such as Prader-Willi and Angelman syndrome in humans, which also express genomic imprinting, with difference that the phenotype or manifestation is different whether the allele comes from mother or from father. Let's now discuss cytoplasmic inheritance. Up to now, all traits and inheritance of characteristics we discussed refer to the genes located on chromosome within nucleus, and Mendel's principles of segregation and independent assortments refer to those traits. However, not all genetic material of our cells is found in nucleus. There are some DNAs located on chromosome within cytoplasmic organelles, particularly mitochondria and chloroplasts. And the inheritance of these traits does not follow Mendelian's principles. And it is because there are particular peculiarities of this inheritance. Firstly, Even though these traits are present in both males and females, they are usually inherited only from one parent, from parent which provides cytoplasm with the organelles. In mammals, it is always mother. So in mammals, cytoplasmic traits encoded by genes localized on DNA in mitochondria is always inherited from mothers. Similarly, as with sex-linked inheritance, reciprocal crosses don't give you the same outcome. And because division of cytoplasm is not so strictly controlled as mitosis and meiosis, there is high phenotypic variation in cytoplasmic inheritance. As we will discuss later, human mitochondrial DNA has circular forms and encodes limited number of traits. There are approximately 37 genes which are localized on mitochondrial DNA. This is a very small number of genes when you compare it to nuclear DNA. However, several of these genes encode components of the electron transport chain important during cellular respiration. So basically, if you remember importance of cellular respiration in generating energy for normal function of cells and well-being of organism, you can imagine importance of these genes. In addition, although one circular mitochondrial DNA is small, each mitochondria possesses approximately somewhere between 2 and 10 copies, and most importantly, one cell contains thousands of mitochondria. So even though one DNA is small, the full amount of DNA within cytoplasmic mitochondria is huge, and it's why it's so much easier to isolate mitochondrial DNA than isolate nuclear DNA. And very often when you read about the DNA isolation from fossil material, it's mostly isolation of cytoplasmic DNA. As we discussed before, one cell contains thousands of mitochondria, and these mitochondria segregate randomly during cell division. As you recall, there is no controlled mechanisms for division of cytoplasm, such as one which controls division of DNA into daughter cells. So this randomness in mitochondrial segregation is behind the extensive phenotypic variation in genes or in traits encoded by genes located on mitochondrial DNA. Let's consider, for example, one cell where random mutation occurs within mitochondrial DNA. So you have wild type and mutated mitochondria. Now, these cells undergo several rounds of division. During each division, mitochondria segregate randomly. So what would happen after second, third, or fourth division? You can end up with cells which would have all either mutated or wild-type phenotype, or you can end up with cells which would have a mix of mitochondria containing mutated and wild-type DNA. So this segregation, random segregation, results in progeny that may differ in their number of mitochondria which possess wild type or mutated genes. And this randomness, I repeat again, is behind the extensive phenotypic variation in traits encoded by mitochondrial DNA. Cytoplasmic inheritance was recognized by Carl Correns at the beginning of 20th century. He performed experiments with 4 o'clock plants, the kind which is outlined right here. He took pollen from plants with white leaves, green leaves, or variegated leaves and crossed them with seed. He pollinated seed from either white plants, green plants, or variegated plant. What he observed was that the phenotype of offspring, all offsprings are here, were always determined by seed, by maternal parents, and not by pollen, by paternal parent. So variegation in this case results from defect in genes which encode the production of chlorophyll inside of chloroplast. So in white plants there is failure to produce a chlorophyll, so chloroplasts are defective. Green has normal chloroplast and variegated has a mix of normal and failed or abnormal chloroplast. So nevertheless, the major point or major take-home message from his experimentation was that it was always a maternal plant which determined phenotype of the offsprings. So it was first recognition of cytoplasmic inheritance, in this particular case, chloroplast inheritance. This observation applies although also into the inheritance of mitochondrial traits in humans. As we already discussed, in humans as well, cytoplasmic inheritance or respectively mitochondrial inheritance is always determined by maternal parents. So gene encoded on mitochondrial DNA are always inherited, or traits encoded by these genes are always inherited from mother. So next we will discuss the inheritance of continuous characteristics and the effects of environment on the phenotype of offsprings. However, before we go there, let's summarize how sex influences heredity. Now, first let's talk about sex-linked, sex-influenced, and sex-limited characteristics. What is difference? Now, sex-linked characteristics refer to the genes or to the traits encoded by genes located on the sex chromosome, physically located on sex chromosome, so they can be located on X chromosome, we talk about X-linked characteristics, or they might be located on Y chromosomes, and we talk about Y-linked characteristics. How they differ from sex-influenced and sex-limited? Both sex-influenced and sex-limited characteristics are encoded by genes which are located on autosomes. So they are autosomal genes which are manifested differently in male and female. So what is the difference between them? Sex-influenced characteristics manifest in both sexes, so they are expressed in both, but their expression pattern may differ. For example, it might be the same allele might be dominant, behave dominantly in one sex and recessively in another. So it is dominant in male, recessive on female, or vice versa, recessive in male and dominant in female. While sex-limited characteristics are limited to one sex, only one sex shows phenotype. Now, in contrary to sex-linked, influence, and limited characteristics, we have another group of inheritance. So let's talk about genetic maternal effect and cytoplasmic inheritance. Both of them can be traced to maternal parents. Both of them are linked to inheritance from mother. Cytoplasmic inheritance and genetic maternal effect. So what is the difference? In genetic maternal effects, the phenotype of the offspring is defined by maternal genotype. So the genotype of the offspring does not play a role in its own phenotypic manifestation. In cytoplasmic inheritance, also it is still linked to maternal inheritance. Genes which are encoded in cytoplasmic DNA are always inherited from one parent, are inherited in humans from mothers. However, the phenotype is determined by the genotype of the offspring. That's a major difference between genetic maternal effect and cytoplasmic inheritance. In cytoplasmic inheritance, you can trace inheritance to mother only. However, the phenotype determines by own genotype. Now, genomic imprinting, in this case, expression of gene is affected or altered by the sex of the parents who is transmitting this allele. It might be active if inherited from one's partner, and inactive, the same allele is inactive if inherited from another parent. So this is a brief summary. So try to summarize, compare, and contrast these characteristics. Let's now talk about continuous characteristics. So what is the difference between discontinuous and continuous? Up to now, we were only discussing discontinuous characteristics, which have only relatively few phenotypes, so they have discrete phenotype, such as in Mendelian inheritance. You remember in peas seed were either green or yellow, two discrete phenotypes. The seed shape was either round or wrinkled, two discrete phenotypes. So there are discontinuous characteristics because there is not continuation, it's either or. Now, not all traits are encoded or inherited as discrete or discontinuous characteristics. There are some traits which show continuous characteristics, and it is a result of situation where many different genes at many different loci interacts to produce one phenotype. So many genes, interaction, one phenotype. Because there are many genes, sometimes we refer to continuous characteristic also as a polygenic characteristics because they are encoded by genes at different loci. Now, be aware of difference between polygenic and pleiotropy. They sound similar, but polygenic, as I mentioned, many gene, one phenotype, while in pleiotropy, it's completely opposite. One gene can affect multiple characteristics. Now, let's move on into next slide where we will discuss a little bit in more details inheritance of polygenic characteristics. Skin color and eye color are two examples of continuous or polygenic inheritance. We know that eye color is determined by at least two different genes. Even more complex situation is with skin color. It is known that there are many different genes at many different loci which interacts to produce final skin color. Now, there are two kinds of melanin which are involved in color of skin. It is eumelanin and pheomelanin. Now, these different genes are involved in controlling their expression their transport, their ratio, their distribution, and so on. So there are many different genes. And that's why when you look on skin color, you don't see discrete phenotypes, but you see continuous variation which follows bell-shaped curve. And how many different phenotypes you can have depends on number of genes which interacts to produce final phenotype. Let's say if you have one locus with two alleles, how many different genotypes you can have? 3. Yes, you can have homozygous dominant, you can have heterozygous, and you can have homozygous recessive. So it gives you 3 different genotypes. If the interaction is dominant recessive, you have 2 phenotypes. If it's intermediate, you have 3 phenotypes. Now, what about if you have 2 loci? If you have 2 loci, in that case, you have three possible genotype power to 2, which means you have 9 possible genotypes, which if the interaction is incomplete dominance, correspond to 9 phenotypes. Now this is what is outlined here in this very simplified table defining skin color phenotypes. We know that situation is much more complex and there are many more than 2 genes in 2 loci involved in defining skin color. So let's say what if there are 10 genes, what possible combination, how many possible combination you can have. So 3 power to 10, I am not going even to calculate it. So you see more genes interact, more genes are involved, more phenotypes you have, up to the point that you cannot distinguish between individual phenotype because there is continuous variation. The expression of some genotypes may be influenced by environmental factors. We already talked before about the effect of environment on the sex development in turtles and alligators. Here it's slightly different condition. In this case, we will focus on temperature-sensitive alleles which encodes products, protein, enzymes, which are active at certain temperature. One of the best example is expression of vestigial mutation at fruit flies. This mutation triggers development of very short reduced wings if the fruit flies are raised in temperature below 29 degrees Celsius. What does it mean? That the enzyme which is involved in wing development is inactive. However, if you put embryos and let develop fruit flies in temperature above 29 degrees Celsius, even though they have this mutation, the wings develop normally. Now, opposite way it works in Himalayan rabbits. In Himalayan rabbits, you see that fur on the extremities is black, but all body is white. And it is because the enzyme responsible for the color is only active at low temperature. And you can actually make a little experiment to prove this when you shave little piece of fur on the back of rabbit and put ice pack on it. After several days, when fur grows back, you will see that it will be black because the enzyme is active only at low temperature. That's why we call this allele temperature sensitive, and they are affected, their expression is affected by environmental changes. Now be aware that sometimes, very occasionally, environmental factors alone without mutation can produce a phenotype that is exactly the same as a phenotype produced by mutation in genotype. An example might be an autosomal recessive mutation in fruit flies, which is so-called eyelash, which produce greatly reduced development of eyes. You can actually achieve the same phenotype in wild-type flies if you expose embryos to sodium metaborate. So in this case, there is no mutation. Environmental factor alone produce the same phenotype as the mutation. It is phenocopy. The last thing I would like to bring your attention to is anticipation. Anticipation refers to a genetic trait that becomes much more strongly expressed in each following generations. So let's say children have stronger and earlier manifestation. Their children goes even farther, much stronger manifestation and much earlier. This principle or this kind of inheritance occurs when the trait is encoded by genes within so-called unstable regions of DNAs or in highly duplicated region which is unstable. And basically this instability is increased from generation to generation. Anticipation. An example is meiotic dystrophy, which is caused by mutation in one of these unstable regions of DNA.